Hey everyone, welcome back to another day in my life as an apprentice for minorities in track sciences. In this video, we'll be covering the homogenization and assay research procedures we use to take our dissection samples and process them into data ready for analysis. Like we talked about last month, our red tide ecotoxicology study looks at the concentrations of brevetoxins in our local shark species. As water temperatures rise and high amounts of nitrogen and phosphorus runoff lead to more harmful algal blooms, it's important to study the impacts of these neurotoxins on our predator species and assess ramifications across the food web to inform coastal policy and keep our waters clean. The first step of processing our tissue samples is homogenization, which means to create a uniform and consistent mixture by breaking down particles. It is exactly what it sounds like, so we use a homogenizer to blend our samples with a methanol solution. Then, a centrifuge uses high rotation speeds to separate the liquid contents from the solid tissue. The liquid, or supernatant, is then transferred, labeled, and diluted, preparing it for the second part of the lab procedure. And now, time to measure. We perform an ELISA, short for enzyme-linked immunosorbent, assay, which is a procedure used to quickly measure brevetoxin concentrations in liquid samples. ELISA assays can be used in a variety of research, along with ecotoxicology studies like ours. First, we pipette in each sample to compare and contrast with calibrations that have known concentrations of brevetoxin, allowing us to create a standard curve we can use as a benchmark. Each sample is run twice to account for data errors and is also ran alongside control and blank samples. Then comes the brevetoxin antibodies, which start a chemical reaction by binding to the brevetoxin and capturing it on the walls of the test well. To let the chemical reaction occur, we leave the test wells alone for 30 minutes. After its first round of incubation, we wash and decant several times to remove any unbound brevetoxin. We then repeat the 30-minute incubation with a separate antibody designed to attach to the bound brevetoxin and make it detectable. Then we pipette a clear substrate solution that indicates the presence of brevetoxin by turning blue. This chemical reaction occurs while we prepare a computer to run the assay using a software called Gen 5. The final solution we add is hydrochloric acid, which stops the chemical reaction and changes the color to yellow. The samples now have 5 minutes to be measured for absorbance using the assay machine, which runs light through the samples and compares it to the calibrators to indicate how concentrated our samples are. Once the machine measures absorbance and calculates brevetoxin concentrations, we get the data ready for analysis. This includes double-checking our results along the standard curve and measuring variance using Excel, rerunning any samples that might have had errors or too much variance between duplicates. That's all for now. Join us next time as we go over more steps in the elasmobranch research and fieldwork process. Bye!